last, since we left here, I almost felt that sort of blissful indulgence that you feel when one of your friends actually asks to see your home movies, or which otherwise would seem a little much, either braggadocious or just kind of impolite to force that on people. But what that indulgence makes you do is actually just allow your conceited nature sort of to come forward and do that, even though the better parts of you say that no, that's not the kind of thing you should do. But you're not probably going to take me seriously with this hat on anyway. I took Laura seriously because it seemed like a nice fashion statement, but Dan, I have to admit, I didn't believe anything that he said. I figure I'm probably more like Dan than Laura, so maybe, who knows. But I'm going to talk not about Magic Zone. I can just keep on making dumb jokes up here. Detect displays.
Check the displays. This one.
Okay. Oh, here we go. I haven't tap danced for a long time, so I'm glad this works. All right, so I'm just going to talk about things that I move around a lot, I guess. Things that my lab has done, hence the home movie sort of reference there, since I left here. And actually, Jeff and I were fortunate enough to get an NIH grant right as my Darwin Fellowship ended. So, I didn't want anything more than to stay here for another 3 years, and I'm serious about that. So I'm very glad that I was able to stay here for 6 years, actually, in the POTOS lab. And it's been a wonderful experience being at OEB. It's informed the way, being in the POTOS lab has informed the way that I run my lab, because I had very sort of curmudgeonly advisors all the way along. In my undergrad and 2 PhDs. And so I thought that was the way that a lab family was supposed to be. I didn't know that you could have funny little events and things that you do when you get publications and just hanging around the table laughing and things like that. So I thought that's what I want to do when I have a lab. So I'm glad that that was able to happen. I didn't know really what to call it, so I just... so it's just basically a quick tour of some of the lab life that just sort of came ex nihilo since I left here in 2009. And so I told you I was going to show you some pictures. So we have field trips, and there's the lab, and it's been fun. And there, it's a very small graduate student with our lab house bench. So I'll just run through a little project smorgasbord. I won't talk about anything that's building on things that I did at UMass, and I won't talk about any UMass collaborations either. Just things that are sort of new since then. There was a collaborator of mine who unfortunately passed away from cancer after a couple of years of my being there. But we were able to put together his last 3 decades, really, but mostly intensely in the last decade, of breeding experiments with canaries in order to understand the within species genetic differences that can lead to differences in behavior. And so I was really glad to be able to work that out, and it just got published last month in Proceedings of the Royal Society. And I'll just show you one of the results here. There are he used 2 breeds of canaries. One are the rollers that are bred for singing low-pitched songs that people who like song canaries like. And then there is another group the boarders that just sing wild-type high-pitched songs. And it turns out that just with one X chromosome, the Z chromosome difference between the 2 strains, you can have this kind of separation. So all of these individual birds here have 25% border genes and 75% roller genes, but because of doing back crosses, he was able to produce genetic groups one of which has 2 roller Z chromosomes, and one has 1 roller and 1 border. Now remember, roller are the low singing and border are the high singing. So just with that one difference, you can essentially recover high-pitched wild-type singing in an otherwise even genetic background. And so these kinds of genetic differences underlying a learned behavior is something that we don't generally think of when we think of birdsong because it's learned. And so I'm kind of excited looking at the integration there, so we're carrying that forward. A long-standing interest of mine is weaver social behavior. I did some of this kind of stuff for my preparation for my graduate work. Now we've established a field site in Ethiopia. Till then, I had never gone to the same place twice. So it was great. It's great to have a place that you can go back to, and where students can go and not feel like they have to, sort of break new ground. So we've been looking at coloniality, for instance. We found that between species, there are several different species that can be in a particular weaver colony. You can see there's lots of nests here. They're polygynous, so one male will build several nests in most species of weavers and try to solicit females to come and inhabit those nests. And one of my graduate students, master's student, Khalida Khan, has looked at coloniality and found, for instance, that if there's a specific, above a specific threshold of density, they will breed, they will display in synchrony. So, and we've developed a measure and simulations of what would happen if they were not paying attention to each other when they display. And so that's something that we've often wondered whether it was the case, because it seems like it's the case. She's also looked at daily time budgets, and when we go into the behavior that specific males do, we can see that they have certain trade-offs with each other, and different males are in different places in this trade-off. And so, for instance, whereas it's generally considered that males that steal, you can easily see weaver males stealing nest materials from others. These aren't building materials that they're stealing. They sabotage or they build. The males who are stealing from others are not building their own nests, and males that are, or additional nests, and males that are building are generally not taking from others. So that's kind of an interesting, and we have several of these types of correlations that we're looking into now. But this group, there's about 5 undergrads working with her now on weaver song, and I'll see if I can get this, yeah, good, go over there just to show you this macroevolution of weaver song. It's very interesting and rattly kind of thing. This is a typical one, but from a, from a Ruppel's weaver. So analyzing that baby is going to be difficult. But another person on the Weaver project is Bobby Habig, who's just recently left for Beth Archie's lab in Notre Dame. But he's still working on this project anyway, and he's interested in what happens when there's a colony-wide fleeing event, which is a really spectacular thing when this happens, and hundreds of individuals can leave a huge tree. But not everybody leaves, and he found that whether a male leaves when there's an intruder to the colony, and there's this sort of snap with a bunch of males making a click at the same time, and then a whole bunch of them leave. Whether they leave and how long they stay away are both related to how many females the male has. So if he has more females, he'll stay on the colony. And so that's kind of interesting. And he's looking into economic sort of game theory to try to figure out why that would be the case and whether they're doing what we would predict. Speaking of weavers, an interest of mine, and no students are involved in this, I try to keep one little part of my weaver work just to myself, and that would be macroevolution of defensive strategies. Some weavers, when cuckoos come in to try to lay eggs in their nest, mob them, like this lesser masked weaver, and other ones have broadly diverse egg color, so they essentially allow themselves to be parasitized, and then they look at the eggs to see whether it resembles their own. And we're using tetracolor space now to look at, because birds have 4 color receptors, cone types, and so we can look at the way that females can look at their own eggs to try to determine whether they have been parasitized or not. But why do these weavers differ on a macroevolutionary scale? So whereas in my graduate work I looked at one species intensively, now I'm looking at why these things happen between species. So for instance, here's 3 species, and if you look at a whole bunch of different traits that are effective deterrents or responses to brood parasitism, you get some traits that are used very effectively in certain species, and other ones that are used to some effectiveness, and other ones that are completely useless. So for instance, egg color, population or individual variance, and egg rejection is used in the village weaver and used to somewhat less effectiveness in the Ruppel's weaver. The lesser mass weaver has no variation at all. All their eggs are white. They rely instead on prevention. So they have a long, narrow entrance tube. They guard the nest much more than the other ones do, and they desert the site of the cuckoo. So what this has suggested, I was looking at 2 alternative hypotheses, and I was just using these 3 species to begin this study, because in our study site there are 9. And we found evidence essentially for both. And one of the alternatives is that there's only one trajectory of coevolution with the cuckoo, and all the species are at a particular point on that trajectory. That seems to be the case between the village weaver and Ruppel's weaver. And the other alternative is that there are different qualitative strategies that different birds might adopt, and that seems to be the case between the lesser masked weaver and the other 2. So then the question is, why the difference here? So we'll be looking at that in the future. I mentioned eggs. I've been interested in why birds' eggs are colored the way they are for quite a while. I'm trying to develop a general theory of that in terms of, here's a Venn diagram of the costs that would lead to selection on egg color. And one of my master's students, Johanna Navarro, is looking at the evolution of egg color. The first half of her master's thesis was looking at the effect on blue eggs of sunlight. And so we found that and this is going to throw a wrench into some museum studies of eggs and some wild studies too over the incubation period. Actually, the average reflectance of blue eggs goes down after hours of light exposure. And this has been known to happen over a long period of time in museums, but it wasn't known whether time or light is the factor. And so since it can happen just in 108 hours, it's definitely the light. But we don't know what the mechanism is, and so we're collaborating with some people at SUNY Forestry School who have infrared cameras and things like that, can look at the mechanism of the biliverdin change. The second half of her project is on the evolution of eggs in our most ancient lineages of birds, the ratites and tinamous. So you have an emu egg right up here, you can look at those. And then there's an ostrich. And she's holding a tinamu egg right there. Tinamu eggs, if you've never seen one, are just amazing because of the gloss. So even though there aren't very many species in this ancient lineage of birds, and they have a different jaw than all the rest of the birds, they're very unusual group of birds. They have a high variation and very unusual characteristics in their eggs. And so we are looking at the evolution of those. PhD student, my lab thought this was too controversial of a picture to put here because he looks like he's some kind of savior or whatever, but in fact, and they're not really his assistants either, they're just buddies of his over there. And in India, where my PhD student Aaron Owen has been for the last 9 months on a Fulbright fellowship studying rapid evolution of sexual traits in the dwarf or small Indian mongoose. And the thing about this animal, just like the weavers that I studied for my PhD, they were introduced to a lot of different places. So he's in India right now. He's already done work in Hawaii, and we just got word 2 days ago that he's got the funding now to go to Jamaica and Puerto Rico, and we're still waiting on word about going to Fiji after that. So he gets a nice winter too coming up. And the reason why this is a good species to study is because he's interested in the evolution of sexually selected traits, and he wants to do that in sort of real time or historical time. And we found that the anal pad area is, so this anal pad is like a paintbrush that distributes the scent glands from the scent from the anal glands, which is generally either between species or within species among males, and is thought to be a sexually selected trait. And sure enough, the anal pad area is sexually dimorphic and scales with body size in or increases faster than body size in males, but not in females. It's also condition dependent in males and not in females. So in males, but not females, better conditioned males can have larger anal pads. And so what we're suggesting is this is a good candidate for a sexually selected trait, and we expect, based on previous research by Aaron and by others, that whereas the home ranges in India are really broad and they don't tend to overlap in their solitary and their social behavior, we expect to see, and we did see in Hawaii, increased social behavior in the introduced populations. There are no predators in any of these places. They're islands, and they have plenty of food. And so we expect them to interact more with each other, and then possibly this will, and sexual selection theory predicts it will, lead to more intense sexual selection, which means we expect to see an evolution of sexual dimorphism and evolution of the anal pad. So we're going to see whether that's the case. The data is already in on that because he's just finishing up in India, but we still haven't looked at it to see whether it's the case. All right. Those other projects notwithstanding, the most significant project in terms of activity in the lab is our cultural evolution project, and we're using the house finch for that nice bird that's common in New York and was introduced to New York in 1940. And I will just play a little warble of that. This is what I hear when I walk into my office every day, because this individual was actually recorded on Queens campus. And that was recorded, as you can see, by Franny Geller, who's another PhD student of mine. And as I alluded to, the reason why this is a good species to study is not only because it has a complex learned song and it passed across generations by social learning and modified as many birds do, but it was introduced from a stable native range in California and in the West in general in 1940 to New York City, and from there has spread throughout the entire eastern seaboard and is now as far as west as the native range is. And we have historical recordings from throughout these areas historically. So we can do both, in Andrew Hendry's terms, synchronic and allochronic studies of cultural evolution. So we can do it spatially and temporally. And so we have been going all over the place. I have one master's student in Hawaii right now, and one just came back from California, and one's going to New Mexico. And we're trying to record these birds now all over the place so we can compare then and now, and then compare spatially also. And Franny herself is mostly interested in the single link of one generation. What is the mechanism within one generation that leads to, when you concatenate it, cultural evolution over the generations? And so you can see right here, for instance, one individual male will sing 3 different songs, and then you'll see yearlings hanging around with that male. And this yearling, for instance, in March sang something as a chunk that resembles this part of this song of the adult that he was hanging around with, and then in April it resembles it even more, and then later on was nearly identical to it. So we're learning that, for instance, that males learn from each other in chunks, and that they hang around with the male preferentially whose song that they have learned, and they modify it over the course of their first spring, and all these kinds of things. And this is essentially one step in cultural evolution. This is what's going on. And so if we can multiply this across the population, across time, then we can understand what's going on. So we'll take individual syllables and we'll string them out just like you would a protein sequence of amino acids or a gene sequence of bases, and then we can identify things like insertions and deletions, other kinds of cultural mutations in the population. But this leads us to a computational problem, and another PhD student of mine, Chen-Hui Zhu, has just developed her software, Finch Catcher, which allows us to analyze large numbers of songs and more objectively than just looking at the songs and trying to see, is this the same syllable as this, or has it changed or not? And so by doing so, she was able to put together huge trees and do a massive alignments of thousands of songs. I'll just give you one example. This is about 4,000 songs from 8 localities, 240 individuals. And she produced 52 syllable types among those, and you have the clade of localities here. So the bird originated in Mexico, and moved up into California, and we already know from historical records that the New York birds are descended from the California birds. So this clade here that's developed purely on these cultural traits agrees with the historical and genetic data there. And we can see that New York then and now are pretty similar with respect to each other. This is a heat map, so the red bars mean syllable types that are very common in that particular population, and the blue ones are rare. And so you can see that there's some sharing there between the two New Yorks and between them in California. But then the other populations are very distinct from each other. Now, we also have a theoretician in the lab. It's sort of a chain or it's a little pretentious, but he really does this in Brooklyn, before drafting. But Elliot Aguilar, another PhD student of mine, is looking at the theoretical predictions that we would make once we get this broad scale of cultural evolution in the house finch. And the way that he's doing that is to apply something that's been used in genetic evolution, which is coalescent theory. So we'll start from today and we'll work back and see how, based on different things like population size and the learning rate, alpha, which is how many individuals does one individual's song end up getting transferred to through the learning process. And by so doing, he's been able to predict, just on a neutral theory right now, he's bringing in selection for the last half of his PhD, but based on just neutral expectations, based on learning rate, population size, and number of individuals who have the particular trait or syllable type or whatever, and this could be applied to pottery styles or fashion or anything else, how far you would expect you would have to go in generations in order to find the most recent common ancestor of that particular syllable or trait. We're dealing with problems like with homoplasy right now, that syllables can have multiple origins, that kind of thing. All right, now I told you I skipped OEB collaborations, but I do value very much the continuing collaboration. Some of these are more active than others, but one active one is with Jeff and Dana, of course, still on the swamp sparrow stuff, and with other members of the POTOS lab on performance analysis. Jeff is just getting finished with some of that. Steve Johnson, a former graduate student here, on analyzing sequences or syntax in our house finch song based on his stuff on robins. And then Norman and two previous Darwin Fellows, Andrew Hendry and Ted Stankiewicz. We've been working on relaxed selection and trait loss for years. We slowly produce papers, about 1 every 2 years on that. And then with Dan, we're looking at bird egg color and thermal regulation, sort of where our interests coincide there, and we're coming to the close on that as well. And some of these, all of these papers have come out since I left here on these collaborations, so I'm really happy about that. So there's the lab. Thank you very much.